Dr. Gonzalez-Lima, this is an absolute pleasure. I told you I've been looking forward to this conversation for a long time. Welcome to the show.
Thank you.
Let's get started with I would love to hear how you got so fascinated with the brain because you've been in the field for a little while, but what sparked the interest in the brain specifically?
Yes, when I was an undergraduate at Tulane University in New Orleans, I had a wonderful professor, Dr. Joanne King, that in one of the classes, she brought a human brain that had been fixed, and she started dissecting the brain in front of our small class. And we were able to take a closer look at the structures in the brain. And she was just a brand new professor, very excited, motivated. And at the time, I was interested in doing an honors thesis project, and this sparked my interest in exploring the brain.
Very cool. And so from there, the rest is history. And now you're leading so much research in the neuroscience field. When it comes to a lot of your work, I think just to give people a foundational base, um, mitochondrial respiration,
Mhm.
why should people care in terms of
Yes.
foundationally, why should
Uh,
they care?
the key to the work that I've been doing for many years is how neuronal function is maintained.
Hmm.
Neurons, brain tissue in general, uh, is a high demand organ in terms of energy use. So, for example, uh, more than 95% of the oxygen that we breathe is used by mitochondria. And
Hmm.
mitochondria are inside the cells, and they use that oxygen in a process that is coupled with energy production in what they call ATP, the adenosine triphosphate. But the key to understand this is really the key to life on the planet, because most life on the planet depends on the use of oxygen.
Hmm.
And actually, what mitochondria do is that they have a reaction that transforms oxygen into water. And that reaction then is coupled to adding a phosphate in this process of so-called oxidative phosphorylation. It's
Hmm.
called oxidated because it's based on oxygen. And what that does is that oxygen that was bound, when it breaks away, it releases heat. This heat is what we refer to as calories. So when somebody eats food and you say that the food is used as a fuel for energy in your body, what you're saying is how many calories, how many of these phosphate bonds can be broken. And when the phosphate bonds are broken, they release a small amount of heat. And if you remember in basic chemistry, there are many reactions where you have a lot of reactants that are together, but nothing happens. And then you provide heat to the beaker where the reactants sit, and then all of a sudden products happen, a
Hmm.
reaction takes place. This is exactly how all of the biochemistry in the body that has to do with energy is controlled. The reactants are manufactured, they're available there, but nothing happens until you break down this ATP, you release heat. It's like putting your burner on, and then the reaction takes place, you get a product.
Hmm.
So the brain depends all the time in this process because the brain doesn't store energy.
Hmm.
So the energy depends on you constantly having availability of oxygen and being able to use the oxygen to generate these ATP molecules that then can release calories. Uh, So the mitochondria is at the center of being able to maintain your brain essentially plug in, being able to function.
Amazing. Thank you for the explanation. Uh, there are a couple of particular, I guess you would call them either chemicals and or therapies that I would love to just drill down on with you today. But I found one commonality among these two, something called cytochrome oxidase. And though we may have addressed it a little bit on the show in the past when it comes particularly to low-level laser therapy, but do you mind just for the uninitiated, cytochrome oxidase,
Yes.
what is it?
Yes. Well, let's start with the name, cytochrome. Cyto means cell, and chrome means color. So this is actually a molecule that gives color to the cell. But what does it mean to give color to the cell? Uh, what color means is that certain wavelengths of light are absorbed, but others are reflected. So this cytochrome oxidase is the so-called main photoacceptor, that is, accepts light. Light is made up of photons, so it accepts photons. And why is it called oxidase? Oxidase comes from oxygen,
Mm-hmm.
and it is an enzyme. An enzyme is a biological catalyst. A catalyst is a chemical that is like a matchmaker. It doesn't, it's not part of the wedding, but it makes the wedding possible between different, different chemicals. And so what it does is it allows, you remember that I mentioned oxygen that we breathe have to be reduced into water.
Mm-hmm.
Well, cytochrome oxidase is the catalyst that has to be there for that matchmaking to happen.
Mm-hmm.
And so that 95% of oxygen that we consume that is used for energy production is all used or consumed in that reaction that is catalyzed by cytochrome oxidase. So without cytochrome oxidase, it doesn't matter how much oxygen we have, we cannot convert that oxygen into a process that will generate energy.
Mm-hmm.
So this enzyme is crucial to what is referred to as aerobic life, life depending on air, by air meaning having oxygen in the air. So all organisms, all cells that use oxygen or air have this enzyme. So from microbes to lower organisms to us. And this enzyme is inside the mitochondria. So
Mm-hmm.
it is a mitochondrial enzyme. So that's the key to cytochrome oxidase. So if your brain is working harder, processing more information, it uses more energy, so it means there is more cytochrome oxidase activity.
Mm-hmm.
However, if you demand more from your brain that it can provide at a given time, you're going to need more energy to sustain that. So this enzyme is highly regulated. It can be so-called upregulated on demand.
Mm-hmm.
And in fact, it can be downregulated on demand. If you don't use your brain,
Mm-hmm.
your ability to generate energy also goes down. So the advantage of this enzyme is that my first interest was using the enzyme as an index of brain function,
Okay.
because oftentimes you use as an index of brain function the amount of oxygen that is consumed or the amount of blood flow bringing the oxygen to the tissue. But this enzyme is the most direct connection to neuronal function because it directly relates, is coupled with this neuronal activity.
Can the average person measure that, or is this something that I have to make myself a guinea pig and go into a university lab in order?
Yes, for many years, it was not possible to measure this in a non-invasive way
Mm-hmm.
I had to extract brains from animals and freeze the brains and then process them through what is referred to as a histochemical
Mm-hmm.
procedure, a procedure where you can reflect the chemical reactions by in the tissues. And in fact, in the early 90s, I developed in my lab the first quantitative histochemical procedure to be able to measure cytochrome oxidase activity uh, in brain tissue.
Mm-hmm.
And this opened up the opportunity for us to monitor this enzyme. But the experiments were very difficult because you always have to, at the end of the experiment, you have to take the brains out
Yeah.
and measure it. So it was not until very recently that collaborating with bioengineers, uh, we have been able to measure this non-invasively
Mm-hmm.
uh, by putting sensors on the foreheads of people
Okay.
uh, in a way that in a procedure that we call broadband near infrared spectroscopy. It
Mm-hmm.
follows the similar principles that we did with the actual tissue in the lab,
Mm-hmm.
but uses near infrared light that can go through the forebrain uh, and in a very broad range of wavelengths, because all molecules absorb light at different wavelengths. And you remember that I told you that cytochrome oxidase was the primary photoacceptor that we have inside the brain
Mm-hmm.
or inside cells, actually. Uh, so it allows us to know from all the light that we provide, it allows us to measure how much of that light is absorbed in particular wavelength. And with that, we can calculate how much cytochrome oxidase concentration is there through the forehead in the brain. So we've been doing that only recently. Our first publication was 3 years ago in
Mm-hmm.
2017. And before that, we weren't able to measure this. So now we can do it non-invasively in people, but it's not something that is available beyond. There's probably our two groups in Texas, and there is also another group in University College London.
Mm-hmm.
And there used to be one in Berlin, Germany, but it hasn't been active recently. And all of these are these very few places where they've been able to develop independently uh, a way to measure non-invasively from the human head cytochrome oxidase.
Sounds like I need to make a trip to Austin. This is fascinating. And now I want to come back to the histochemical process that you developed in a little bit, because I think in your May 2020 paper, there was at least when you were working with rats, you
Correct.
brought that back.
Yes.
Um, we can come back to that in a little bit if we have time. But with cytochrome oxidase,
Mhm.
and forgive me, this is a fairly elementary question, but over time, is it something that dissipates? Like, could it be a marker for longevity, for instance?
Mhm. Yes, cytochrome oxidase, yes, it is very tightly related to longevity.
Mm-hmm.
If you can upregulate cytochrome oxidase, you upregulate the lifespan. So, for example, very simple animals like insects or like a fly that lives for a very short time, if you are able to regulate cytochrome oxidase inside their mitochondria, uh, you can measure that they can live longer.
Mm-hmm.
And the main reason for this is can be it has to do with two factors. One that I already mentioned, that you can use oxygen to produce energy, so energy is available for the organism. But number two, which is very important for longevity, is the process of using oxygen. Remember, I told you involves the oxygen being reduced I mean, in chemical terms, is transformed into water.
Mm-hmm.
Reduction means the oxygen binds to hydrogen and it becomes water. So water is non-reactive, is in a neutral type of a state. And this is the ideal situation that this reaction catalyzed by cytochrome oxidase. But as you do this reaction, there is always a small fraction of the oxygen that is not fully reduced into water, that is not fully neutralized, that is, it still have some electrons around its orbit that want to react with
Mm-hmm.
other compounds. And this is what is referred to as reactive oxygen species.
Mm-hmm.
These reactive oxygen species are primarily formed there in the mitochondria in this process of oxygen consumption for energy production. So the more efficient cytochrome oxidase in mitochondria is to fully reduce oxygen into water, the less reactive oxygen species you have.
Mm-hmm.
And these reactive oxygen species is what are also referred to as the main class of free radicals.
Mm-hmm.
They want to neutralize this free electron by binding, sharing that electron with some other compound nearby. And by doing that, they create what is called oxidative stress. That is, they're oxidizing molecules like DNA or
Mm-hmm.
proteins. First, the ones that are immediately around them inside the mitochondria. Then they oxidize the ones that are in the membranes of the mitochondria, the lipids membranes. And then they form so-called reactions by oxidizing one molecule, sharing an electron with one molecule, they push and create like a chain reaction that is called a lipid peroxidation chain.
Mm-hmm.
So all of these leads to what is called oxidative damage.
Mm-hmm.
I need to clarify something here that is often confused. Oxidative stress by itself is not entirely a negative thing,
Mm-hmm.
because when you have oxidative stress, that serves as a stimulus to create antioxidant enzymes. In other words, ways that the body can neutralize that. And the whole point of the neutralization of the reactive oxygen species is to make them into water. So
Mm-hmm.
you go through a cascade of reactions that they get closer and closer to water. So which one is the number one antioxidant enzyme? People often don't know this. The number one antioxidant enzyme is cytochrome oxidase, because it's the one that is capable of converting oxygen fully into water. So cytochrome oxidase then does two things. It allows you to derive energy from oxygen in the process of oxidative phosphorylation inside mitochondria. And it has an antioxidant action at the very beginning
Mm-hmm.
of the reactions of this reaction that have to do with oxidative stress. So by allowing these two phenomena to go hand in hand, you can prolong the life of the cell, and you can prolong the life of the organism.
This is fascinating, and you just eloquently walked us through some pretty complex topics of biochemistry. So thank you very much. Where did your interest in methylene blue come into play, or how did you hear about methylene blue?
Yes. So first, we came up with a way to measure cytochrome oxidase.
Mm-hmm.
We use that for monitoring brain activity. And then in the process of our research, we clearly saw that when animals were exposed to complex or even simple learning tasks, learning and memory processes, there was always an upregulation of cytochrome oxidase.
Mm-hmm.
So I became interested in then, so what happens when people have memory deficiencies? Is there a phenomenon that is related to this use of oxygen for energy production? And one of my graduate students and I embarked in an ambitious project. So the only way we could answer this is by doing these histochemical reactions in very fresh brains from
Mm-hmm.
humans. So the one place in the world that we could do this is called Sun City, Arizona.
Oh, I thought you were going to say South Africa there for a second, but
No, no, no. This is a retirement community. Is a city only for retired people.
mm-hmm.
The advantage is that they all belong to the same healthcare system, the Sun City healthcare system, and they are all people who suffer from a number of these diseases of all age. So they are very generous in donating their brains and organs for research. So we went there and spent a summer literally working with a pathologist. Whenever somebody was going to pass away, the pathologist and I, my graduate student, will go there and he will declare the person dead, and then we will have a saw and go zzz, remove the scalp and get the brains out, and fresh brains, and freeze them.
Mm-hmm.
They were interested in collaborating with me because as a neuroanatomist, I will be able to get this brain and dissect the brain into small pieces and label all these pieces. And then I will have one piece for me and another piece for the research program
Mm-hmm.
there at Sun City. And I will store those there. We spend the summer, and also we use the brain bank that they've been storing and dissect them.
Mm-hmm.
So we make a unique sample of fresh frozen brains from people with Alzheimer's disease and people who were controls. That is, people who had the same age and other characteristics, who were living there in the same community. And oftentimes we even have the spouses of the ones that had the Alzheimer's. And we analyze this using various techniques. All of the techniques that are used for staining for all of these changes that happen in the brains of people with Alzheimer's, in addition to this quantitative histochemistry. And
Mm-hmm.
what we found that verified our animal experiments was that the primary deficit in the fresh brains, not in this fixed tissue that is used for other analysis, but in the fresh brains,
Mm-hmm.
was a down regulation or inhibition of cytochrome oxidase in the brains of Alzheimer's patients.
Wow.
Since then, this is how I got to methylene blue. Since then, I was interested in, okay, now we know this. This paper came out in 2001. Very highly cited paper, because no one has been able to come up with a sample of fresh brains that are so well controlled like this. And to also clarify some issues about Alzheimer's, these amyloid deposits became very popular as the hypothesis for the cause of Alzheimer's. Had absolutely no relationship with the
Really?
cytochrome oxidase inhibition.
Mm-hmm.
So
What
these,
about tau? Because tau comes up in this discussion quite a
yes,
bit
so
as well.
tau is the other pathological sign. The problem with this amyloid and tau, so-called neurofibrillary tangles, the one that have the tau. These things are consequences and not causes of Alzheimer's disease and these neurodegenerative changes. And they, of course, even though you hear them in the context of Alzheimer's, they take place in all kinds of neurodegenerative conditions. And so they are what you see after years of this process happening in the brain. The cytochrome oxidase is at the forefront, is the early event, is the inability of the brain to use oxygen to prevent this oxidative damage. So unfortunately, the idea was that that many people have spent by now billions of dollars researching the amyloid hypothesis. You can clean up your brain entirely of this amyloid, and it has absolutely no beneficial effect on your memory or your dementia or the progression of the neurodegeneration or anything of relevance in Alzheimer's disease. So in these clinical trials, the drugs were very effective at, not at the beginning were highly toxic, but once they spent not millions, not hundreds of millions, but thousands of millions and suffering of people going through these trials, all the experiments were a success. We can clean up all of this, but of course it makes absolutely no difference. And it produces a number of side effects. So to answer your question, the amyloid is at no relationship, is a compensatory mechanism. In fact, at the beginning, is something that you want to happen, not later on. And the tau aggregations, the amyloids happen outside the cell, and the tau happens inside the neurons. And when a neuron has this tau that it has a so-called fibrillation inside, it has been phosphorylated inside the cell, it means the cell is functionally dead. It is a ghost. It's just like a body that is sitting there and it still hasn't been removed from your brain, but is completely non-functional. So when you identify these neurons with the phosphorylated tau inside, those neurons are functionally gone. And you can remove the phosphorylation at this point, and it is like trying to resuscitate an organism that is already dead. So it is too late. When you see that, those are no longer reversible. So stopping this chain of biochemical change that has to do with tau phosphorylation doesn't do anything, because those neurons cannot use oxygen, cannot produce energy, so they cannot function. So again, all the efforts are directed towards that, and or that believe that that's what the mechanism will be for improving neurodegeneration are not going to work. Unless by mistake, which is what happens with methylene blue, they think that they are acting on the tau phosphorylation as the primary event, but in fact, they are producing other effects on cytochrome oxidase that are the benefits, the beneficial ones. So how methylene blue comes into this picture?
Yeah.
I was trying to find, I also teach pharmacology and toxicology, trying to find, is there any compound, is there anything that I can use to facilitate cytochrome oxidase activity? I know it normally, if you stimulate your energy use, then the enzymes upregulates. So if you use intellectual challenges, your enzyme builds up in your brain, just like when you do exercise. In fact, when you do aerobic exercise, the enzyme upregulates inside your muscles,
Mm-hmm.
and then your muscles can more effectively use oxygen. So to give you an analogy, if you start running and you think that you run out of air, uh, you go like, and it doesn't matter how much air you come in, you cannot generate more energy, is because the limiting factor is the amount of cytochrome oxidase.
Mm-hmm.
So you're putting more air, your circulation is going, your heart is pumping blood to your brain that is oxygenated, but the limiting rate is cytochrome oxidase. If that oxygen cannot be reduced into water, you cannot generate more energy. So how can we accelerate, go around that process?
Mm-hmm.
And I found methylene blue. Methylene blue was the key to this process.
Mm-hmm. So methylene blue, if many people out there may not actually know what it is. In my research, this chemical has been around since I think the 1890s and was once used for malaria. It's used in cleaning fish tanks. It's got a whole heck of a lot of uses. Do you mind just
Yes.
going through? I would love to hear just more about methylene blue as a compound, and
Okay.
then how you kind of broke it down pharmacologically to be
Mm-hmm.
used in your research.
Yes. Uh, Methylene blue is it's an exciting thing. It was developed not in the 1890s, in the 1870s,
Oh.
1876, by a German chemist. This was developed at the beginning of what is called the Industrial Revolution. The
Mm-hmm.
first thing in the Industrial Revolution were the textiles, the companies that make clothing, and they used natural dyes. And the chemistry was advancing rapidly at the time. So they synthesized some chemical dyes. And the first of the blue ones that was synthesized was what eventually became known as methylene blue. The most common name at the beginning was methylthioninium chloride. Uh, and it belongs to a family of compounds uh, that that are have been related to they're called phenothiazines
Mm-hmm.
that have been related to other compounds that are common nowadays, but methylene blue was the parent compound, the
Mm-hmm.
first one that was synthesized of that family. So why it was synthesized? Because it reflected blue light. You remember the cytochrome oxidase I told you accepts photons and reflects? So methylene blue has this property of absorbing. It absorbs red type of photons and reflects the blue types.
Mm-hmm.
That's why it's blue. Uh, it was used in the textiles, for example, I see your shirt there that is bluish.
It is, yeah. Mm-hmm.
So they will use it for that, for the blue jeans. The
Mm-hmm.
blue jeans were dyed with methylene blue. In the 1800s and 1900s at the beginning, methylene blue was widely used in industry. But then in 1890, because there was a scientist in Berlin called Paul Ehrlich, and Paul Ehrlich was the first one to inject methylene blue, this synthetic chemical, inside a living organism. He injected in a rat intravenously, and the rat was alive.
Mm-hmm.
And then he dissected the rat and found out something that was very surprising at the time, that you could inject a chemical systemically that is going through the circulation throughout the entire animal, but that it was selectively staining nervous tissues. So the brain and the peripheral nerves, where the nervous tissues were, they became stained.
Mhm.
And this led Paul Ehrlich to use the term magic bullet. This is where the term magic bullet came from.
Oh, really? From methylene blue?
The methylene blue, the original magic bullet. And why was it a magic bullet? Because you threw that bullet into the circulation and it went all over, but target the nervous tissue. What he didn't know was the reason it was targeting the nervous tissue was that it was being concentrated in mitochondria. Because the mitochondria are the most active parts of the inside the cell that have to do with respiration, with oxygen consumption. They are more highly concentrated inside nervous tissue. The second order tissue is, the heart, the muscles that are very active, but the number one is nervous tissue. So this is actually a methylene blue is actually a mitochondrial stain, but the organism has to be alive when you inject it. So there have to be oxygen consumption happening, and methylene blue finds its way to this exchange of electrons. You remember that I told you about oxidative phosphorylation and cytochrome oxidase? Well, the whole process of how the food that you eat ends up in calories is because the food that you eat is transformed in what are called electron donors. And the electron donors donate an electron to the electron transport chain, and that electron moves along several molecules inside mitochondria until the last one, that is cytochrome oxidase, which cytochrome oxidase is the only one then that can donate that electron to oxygen. Oxygen is in nature, is the ultimate electron acceptor. That's why the process of removing electrons from a chemical is called oxidation. It's named after oxygen. That's what oxygen does. It removes electron. It oxidizes compounds. So this phenomenon of the electron transport, normally we get the electrons from the food that we eat through the electron donors. Everything that we eat becomes basically two electron donors types. What happened with methylene blue? When methylene blue at low concentration has a property that I have not found in the same way in any other chemical after 120 years of synthetic chemistry, it auto-oxidizes, and at low concentrations, it maintains cycling. So it donates electrons and it grabs electrons from its immediate environment. So methylene blue finds a reaction when this is happening, and this is happening primarily inside mitochondria in the electron transport, and then it becomes an electron donor to the electron transport, and that electron goes to oxygen. So it accelerates oxygen consumption as if it were part of the machinery of the oxidative phosphorylation. Wow. By accelerating oxygen consumption, you accelerate your energy production, and you also act as an antioxidant. And remember, because it not only donates electrons, but it can also accept electrons. So if there are any reactive oxygen species that are not fully reduced, methylene blue can directly interact with them as an antioxidant. So then methylene blue becomes like an artificial electron, I call it electron cycler, because it not only donates, but it can accept electrons if there are too many. But it has to be a low concentration. In low concentration, it maintains this redox cycle. In chemical terms, is neither reduced nor oxidized. It maintains a net redox state that allows it to continuously provide this. So this is how methylene blue can then affect energy production, antioxidant processes, how methylene blue can prolong life. If you give it to that fly that I told you, that fly will live longer because of the methylene blue providing energy and preventing oxidative damage. But it has to be at this low concentration. If you give it at a high concentration, it becomes primarily an oxidating agent. It grabs the electrons from oxygen, and it doesn't allow oxygen then to be formed, and that doesn't facilitate then the energy. So that's where methylene blue comes to this picture. But
Dr.
why do you say, you may ask, why methylene blue has been used for so many things? Because this process of electron cycling is so, it's part of redox chemistry. All kinds of reactions, chemical reactions have to do with this exchange of electrons.